Alright, the chapter 1, the animal cell. All the tissues and organs of which the body is composed were originally developed from a microscopic body, the ovum, consisting of a soft gelatinous granular material enclosed in a membrane and containing a vesicle or small spherical body inside which are one or more solid spots. This may be regarded as a perfect cell. Moreover, all the solid tissues can be shown to consist largely of similar bodies or cells, differing, it is true, in external form, but essentially similar to an ovum. In the higher organism, all such cells may be defined as nucleated masses of protoplasm of microscopic size. The two essentials, therefore, of an animal cell in the higher organisms are the presence of a soft gelatinous granular material, similar to that found in the ovum, and which is usually styled protoplasm, and a small spherical body embedded in it and termed a nucleus. The remaining constituents of the ovum, its limiting membrane, and the solid spot contained in the nucleus, called the nucleolus, are not considered essential to the cell, and in fact many cells exist without them. Protoplasm, also known as the cytoplasm, is a material probably of variable constitution, but yielding to the chemist on its disintegration bodies chiefly of proteid nature. Lecithin and cholesterol are constantly found in it, as well as inorganic salts, chief among which are the phosphates and chlorides of the alkali metals. Oh, calcium. And calcium. It is of a semi-fluid, viscid consistence, and appears either as a hyaline substance, homogeneous and clear, or else it exhibits a granular appearance. This granular appearance, under a high power of the microscope, is seen to be due to the fact that protoplasm consists of a network or honeycombed reticulum, containing in its meshes a homogeneous substance. The former is known as spongioplasm, the latter as hyaloplasm. The granular appearance is often caused by the knots of the network being mistaken for granules, but in addition to this, protoplasm often contains true granules, some of which are proteid in nature and probably essential constituents. Others are fat or pigment granules and are regarded as adventitious material taken in from without. The size and shape of the meshes of the spongioplasm vary in different cells and in different parts of the same cell. In many fixed cells, epithelial cells for example, the external layer becomes denser than the rest, and often altered by the disposition in it of some chemical substance, so as to constitute a membrane which encloses the rest of the protoplasm and forms the celly wall. The relative amount of spongioplasm and hyaloplasm varies in different cells, the latter preponderating in the young cell and the former increasing in amount at the expense of the hyaloplasm as the cell grows. The most striking characteristics of protoplasm are its vital properties of motion and nutrition. By motion is meant the property which protoplasm has of changing its shape and position by some intrinsic power, which enables it to thrust out from its main body an irregular process, into which the whole of the protoplasmic substance is gradually drawn, so that the mass comes to occupy a new position. This, on account of its resemblance to the movements observed in the amoeba or Proteus animalculae, has been termed amoeboid movement. Ciliary movement or the vibration of hair-like processes from the surface of any structure may also be regarded as a variety of the motion with which protoplasm is endowed. Nutrition is the power which protoplasm has of attracting to itself the materials necessary for its growth and maintenance from surrounding matter. When any foreign particle comes in contact with the protoplasmatic surface, it becomes incorporated in it, being enwrapped by one or more processes projected from the parent mass which enclose it. When thus taken up, it may remain in the substance of the protoplasm for some time without change, or may be again extruded. The nucleus is a minute body embedded in the protoplasm, and usually of a spherical or oval form, its size having little relation to the size of the cell. It is surrounded by a well-defined wall, the nuclear membrane, which encloses the nuclear content. These are known as the nuclear substance, also known as the nuclear matrix, which is composed of a homogeneous material and a stroma or network. The former is probably of the same nature as the hyaloplasm of the cell, but the latter, which forms also the wall of the nucleus, differs from the spongioplasm of the cell substance. It is sometimes known as the chromoplasm or intranuclear network, and consists of a network of fibers or filaments arranged in a reticular manner. These filaments stain very readily with certain dyes. They are therefore named chromatin, while the interstitial substance does not stain readily and is hence called achromatin. In some resting nuclei, to be speaking from nuclei which are not undergoing subdivision, the nuclear filaments do not form a network, but present the appearance of a convoluted skein, similar to that found in a nucleus about to undergo division, and which will be immediately described. Within the nuclear matrix are one or more highly refracting bodies, termed nucleoli, connected with the nuclear membrane by the nuclear filaments. They are regarded as being of two kinds. Some are mere local condensations of the chromoplasms. These are irregular in shape and are termed pseudonuclei. Others are distinct bodies, differing from the pseudonuclei both in nature and chemical composition. They may be termed true nucleoli and are usually found in resting cells. The nuclear substance differs chemically from ordinary protoplasm in containing nuclein, in its power of resisting the action of acids and alkalides, in its imbibing more intensely the stain of carmine, hematoxylin, and silica, and its remaining unstained by some reagents which color ordinary protoplasm. Recent investigations tend to show that most living cells contain, in addition to their protoplasm and nucleus, a minute particle with, on account of the power it appears to possess of attracting the surrounding protoplasmic granules, is termed the attraction particle or centrosome. It usually lies near the nucleus. The spherical arrangement or fibrillar rows of granules surrounding the central particle is termed the attraction sphere or centrosphere. These spheres are usually double and are connected by a spindle-shaped system of delicate fibrils, achromatic spindle. They are best seen in young cells which are about to undergo the process of division, a process believed to commence in these bodies. The process of reproduction of cells is usually described as being brought about by indirect or by direct division. Indirect division or karyokinesis, karyomitosis also, has been observed in all the tissues. Regenerative cells, epithelial tissue, connective tissue, muscular tissue, nerve tissue, and probably will ultimately be shown that division of cells always takes place in this way, and that the process of reproduction of cells by direct division is, as some observers believe, merely a sort of imperfect or abnormal karyokinesis. The process of indirect cell division is characterized by a series of complex changes in the nucleus, leading to its subdivision. Hold on a second. This being followed by a cleavage of the cell protoplasm. So are we talking about... We are talking about karyokinesis, right? Okay. Starting with the nucleus in the... Yes, it's back to the question. We are still talking about karyokinesis or indirect division. Starting with the nucleus in the quiescent or resting stage, these changes may be briefly grouped under the four following phases. Prophase, the nuclear network of chromatin filaments, assumes the form of a twisted skein or spirem. I never knew those figures existed. While the nuclear membrane and nucleolus disappear, the convoluted skein of chromatin divides into a definite number of V-shaped loops or chromosomes. Coincident with or preceding these changes, the centrosome or attraction particle, which usually lies by the side of the nucleus, undergoes subdivision, and the two resulting centrosomes, each surrounded by a centrosphere, are seen to be connected by a spindle of delicate achromatic fibers, the achromatic spindle. These centrosomes move away from each other, one towards each extremity of the nucleus, and the fibrils of the achromatic spindle are correspondingly lengthened. These centrosomes are now situated one at either extremity or pole of the elongated spindle, and each is surrounded by a centrosphere, from which fibrils radiate into the investing protoplasm. A line encircling the spindle midway between its poles is named the equator, and around this V-shaped chromosomes arrange themselves in the form of a star, thus constituting the mother star or monoster. Metaphase. Each V-shaped chromosome now undergoes longitudinal cleavage into two equal halves or daughter chromosomes, the cleavage commencing at the apex of the V and extending along its divergent limbs. The daughter chromosomes, thus separated, travel in opposite directions along the fibrils of the achromatic spindle towards the centrosomes, around which they group themselves, and thus two star-like figures are formed, one at either pole of the achromatic spindle. This is termed the diaster. Anaphase. This V-shaped daughter chromosomes now assume the form of a skein or a spirem and eventually form the network of chromatin, which is characteristic of the resting nucleus. The nuclear membrane and the nucleolus appear constricted around the equator of the achromatic spindle, where double rows of granules are also sometimes seen. The constriction deepens and the original cell gradually becomes divided. Telophase. In this stage, the cell is completely divided into two new cells, each with its own nucleus, centrosome and centrosphere, which assume the ordinary positions occupied by such structures in the resting stage. In the case of prickle cells, the subdivision of the cell is incomplete. Here, the achromatic spindle threads appear to persist and bridge across the intracellular spaces, constituting the prickles. The series of diagrams by Professor S. Delepine is intended to explain the formation of some of the most important changes observed in nuclei of cells during karyokinesis or mitosis. It is based chiefly on the work of Fleming, Strasburger, E. van Beneden, Rabel, O. Hertwig, Henneguy, and such. A resting nucleus. Nucleolus and nuclear membrane visible. A centrosome is represented near the nucleus. Chromatic filaments much convoluted. Evidence of longitudinal splitting begins to be distinct in several parts. The centrosome has divided. The nuclear membrane is becoming indistinct. These true centrosomes are widely separated, and the space between them is occupied by the achromatic spindle. Two arrows point to the positions which the centrosomes will ultimately occupy. During their passage to these points, the achromatic spindle seems to be within the nucleus. The nuclear membrane has disappeared. mother star, monoster. The nuclear segments, chromosomes, resulting from the breaking up of the chromatic filament into fragments of nearly equal length, have moved towards the equator of the spindle, where they now form an equatorial plate. These segments are all split longitudinally, metaphase. One half of each chromosome moves toward one pole and the other half toward the other pole, being guided towards the centrosomes by the achromatic filaments. Segments more closely packed and less distinct, owing to the formation of anastomoses. Cell completely divided into two, but bridges remain between them in the region previously occupied by the prickle cells. Alright. I will skip over this process, hence it is only comprehensible when you have a picture at hand, but we are more or less ready with the process of mitosis, be it direct or indirect, and we will now be going to the nutritive fluids.